Okay, we're going to continue our session with a talk by Dr. Robert Ritter, who is going to discuss gut peptides and the control of eating behavior. Dr. Ritter is professor of integrative medicine, integrative physiology and neuroscience at the College of Veterinary Medicine at Washington State University. Dr. Ritter investigates neural and endocrine controls of appetite and body weight, and is especially interested in how signals from the gastrointestinal tract and body fat cells alter food intake and metabolism by modulating neural signaling in the hindbrain. Dr. Ritter is very interested in how neurotransmitters and hormones trigger short and long-lasting neuroplastic changes in the hindbrain, and how these changes produce alterations in feeding behavior that either favor weight gain or weight loss. Dr. Ritter.
Thank you, Danielle, and it's good to be here. Thanks for asking. As Danielle said, I'm going to talk about gastrointestinal peptides, but with the bent of talking about how they modulate synaptic function of the vagus nerve, and that's where my focus is going to be, and how that affects the control of food intake. As we've already heard, the gastrointestinal peptides are localized in the gastrointestinal tract in specialized enteroendocrine cells, which are scattered in the amongst the cells of the absorptive and secretory mucosa of the gastrointestinal tract from the stomach through the colon. And this is a picture of such a cell, an L cell, in the distal jejunum, and you can see that it contacts the lumen and has a process that contacts the extracellular space where secretion of peptide occurs. And this is a nerve fiber passing along in the extracellular space. So we have the opportunity for endocrine as well as paracrine function, as Dr. Moran mentioned. I'm looking for the slide changing.
It's gone.
It's on the pointer. There's just an arrow that goes up.
Right. Well, it's not working.
Someone advance to the next slide.
It's not working.
But we'll advance for you. Don't take the pointer away.
I've got one. Okay, so gastrointestinal peptides are distributed in these endocrine cells, as I said, from the stomach through the colon. And although the identifying actions of some of these peptides were discovered in the early 20th century, 1902 for secretin, 1905 for gastrin, by their actions of increasing pancreatic exocrine secretion and increasing gastric acid secretion. In point of fact, none of these substances were identified as peptides per se until the 1960s and 70s when they were synthesized and sequenced. And we know now that at least some of them, even though there is some dozen or more of these peptides that have been identified, that some of these peptides are involved in control of food intake. And I've listed 4 of the main actors here: CCK, secreted by eye cells in the proximal small intestine, GLP-1 and PYY, 3 through 36, and oxyntomodulin from L cells in the more distal small intestine and large intestine. And then, of course, ghrelin, the sole peptide that increases food intake at this point, which is released from cells in the gastric mucosa. Yes. So gastrointestinal peptides are secreted by these enteroendocrine cells, circulate through the hepatic portal circulation, empty into the systemic circulation, and then they're free to broadcast a signal to any tissue that has an areal, a receptor to these specific peptides, which includes the gastrointestinal organs where they were identified, where they coordinate digestive function beginning with gastric emptying and secretion, pancreatic secretion, and then later by an incretin action whereby a lot of these peptides, including CCK, PYY, and GLP-1, facilitate the secretion of insulin and the assimilation of nutrients post-absorptively. Food intake, which we now recognize as being controlled by gastrointestinal peptides, thanks to the initial work at the Bourne Labs of Jerry Smith and his colleagues, can be viewed as a part of this digestive process, in that it limits the inflow of food into the digestive tract during the meal, and thereby facilitates the efficient digestion and absorption of what's been eaten. And of course, the brain and the innervation of the gastrointestinal tract comprise part of the target array that gastrointestinal peptides act on. So a hallmark of gastrointestinal peptides is that their secretion and their levels in the circulation are controlled by nutrients in the gastrointestinal tract. And here I've replotted data from a study of human subjects that illustrates that when a mixed meal is eaten, there is a rapid and dramatic increase in the blood levels of gastrointestinal peptides in the circulation. CCK is rapidly released from picomolar levels around between 1 and 2 picomolar in the blood, and rapidly rises 6 to 7-fold with the entry of nutrients into the intestine, soon to be followed by increases in PYY 3 through 36 and GLP-1. And in fact, CCK action has been shown to facilitate the release of these peptides in anticipation of the actual direct stimulation of the L cells by nutrients as they move down through the intestine. So the peptides are elevated during the meal, and they can be viewed as one of the signals that comes from the gastrointestinal tract that influences food intake by determining the length of the meal. So that brings me to the point that another hallmark when we think of control of food intake by gastrointestinal peptides is that their action is focused on limiting the size and the duration of an ingested meal. So if we look at these 3 peptides that I focused on earlier, CCK, GLP-1, and PYY 3 through 36, all of them reduce food intake primarily by reducing meal size. They reduce the size and the duration of the meal rather than the number of meals an animal initiates. And you can see here in this example of a reduction of food intake by PYY, that PYY reduces food intake in a dose-dependent manner, and that effect is due virtually entirely to reduction in the size of the meals. Meal frequency is not significantly affected. So what we see here is what the gastrointestinal peptides, particularly those that reduce food intake, have in common in terms of their neural substrate. And Timoran has already referred to this with regard to CCK, but to varying extents it's true of the other gastrointestinal peptides, and that is that these peptides that reduce food intake all increase vagal afferent activity. They increase the firing of the fibers that innervate the gastrointestinal tract. And in fact, their ability to reduce food intake is attenuated or virtually abolished when the abdominal vagus nerve is cut. With ghrelin, the story is more complicated, and I don't intend to talk a lot about ghrelin, but ghrelin seems to antagonize the excitatory effects of some of these neuropeptides on vagal afferent firing, but the role of the vagus in actually mediating the increase in food intake is is not at all clear, and it does not seem like vagal afferents directly mediate the increase in food intake stimulated by exogenous ghrelin administration. Now, for most of my talk, I'm going to focus on the peptide CCK because we know a great deal more about that. It's been studied longer, and we're going to focus on the way in which CCK modulates vagal afferent activity. So I've said that gastrointestinal peptides are controlled by the meal, controlled by food intake, that GI peptides reduce food intake by contributing to the process of satiation, and this is the process that terminates meals. So they're very much a meal-related signal. Vagal afferent neurons participate in this reduction of food intake by GI peptides. So questions that I want to address during the talk are, what are the cellular mechanisms by which GI peptides, and specifically CCK, alter vagal afferent functions? We know that they increase the firing of vagal afferents, but actually there's more to the story that we can add to that now. And then secondly, what mechanisms enable vagal afferent function to be modulated by non-GI signals? Okay, and I'm going to come to that. Non-gastrointestinal signals, metabolic signals, alter food intake in ways that appear very similar to those that we see with gastrointestinal peptide administration. And we'd like to know whether there's a connection between these two mechanisms. So in the first part of the talk, I'm going to examine this question from the vantage point of work, mostly of work that's gone on in my lab during the past few years, and focus on the modulation of vagal afferent synaptic transmission by CCK. Okay, vagal afferent transmission, vagal afferents are involved in reduction of food intake by CCK, in part because they activate vagal afferents. And vagal afferents all release the neurotransmitter glutamate, glutamic acid, amino acid glutamate, in the hindbrain, and that's their primary mode of synaptic transmission. They excite postsynaptic cells by releasing glutamate. And so perhaps not surprisingly, CCK reduces food intake, and that reduction is sensitive to antagonism of the glutamate receptors in the hindbrain. And in the case of CCK, it's especially sensitive to antagonism of NMDA-type glutamate receptors. And this is a very interesting effect for reasons that I won't get into in detail. But you can see here that two different NMDA receptor antagonists, a competitive and a non-competitive antagonist, injected directly into the hindbrain, reverse or prevent the reduction of food intake by exogenously administered CCK. And if these substances are administered by themselves in an appropriate manner, they'll actually can be shown to increase meal size themselves. So CCK, as Tim Moran has already mentioned, activates vagal afferent fibers coming from the gastrointestinal tract. But an interesting thing about vagal afferent fibers from an electrophysiological and cell biological point of view is that they are high probability of release neurons. And that means that they very quickly release all the neurotransmitter they have available and begin to fail in their ability to transmit information from the gut to the brain. This data that I'm showing here is data from a colleague of mine in our department, Dr. Susan Appleyard, who studies the hindbrain in a slice preparation. You make a horizontal slice through the hindbrain and then place it in a tissue bath, and you can record from the nucleus of the solitary tract, where the vagal afferents terminate, and stimulate vagal afferents coming into the nucleus of the solitary tract and drive them in an in vitro preparation. And you can see here, looking at the left panel, which is the only one I'm going to talk about today, that very quickly, while you're stimulating the vagal afferent inputs, that the postsynaptic cell, first of all, starts to fire, but then fails. And And that failure is reversed by the local application of CCK. So CCK, acting on CCK1 receptors, this is an antagonist experiment here, actually enhances the transmission between the vagal afferents and the postsynaptic cell. It facilitates transmitter release and prevents failure. And if you block this ability, as I'll show you, vagal afferent transmission fails and you don't get satiation in response to CCK. Now, the way we think this happens is that CCK activates an enzyme, an extracellular receptor kinase, which phosphorylates a protein in the vagal afferent terminals. And this family of protein is called synapsins. Here we're looking at synapsin 1, but there are 3 synapsins, and they highly conserved group of phosphorylation sites. It phosphorylates the protein at these 2 serines, serine 62 and 67. And synapsins are synaptic vesicle proteins which bind the synaptic vesicles to the cytoskeleton of the neuron, to the actin inside the terminal, and control its availability to supply transmitter for release. When serine 62, 67 are phosphorylated, vesicles are released from the cytoskeleton and increase the availability of transmitter for release in the active zone of the varicosities of the vagal afferent terminals. On the other hand, synapsin is dephosphorylated by a protein phosphatase 2B, calcineurin, and that tends to reduce the amount of phosphorylated synapsin and cause more vesicles to bind and be less available for release. So right at this first synapse, it's possible to adjust back and forth a transmitter available for release from the vagal afferent terminals. Well, CCK, it turns out, increases the phosphorylation of synapsin, and you can see that in this series of panels where we're staining for phosphorylated synapsin, specifically at serine 62, 67. And the quantification here shows CCK rapidly increasing phosphorylation of synapsin, and then it's just as rapidly dephosphorylated. So for about 15 minutes in the rat, synapsin is phosphorylated in response to CCK, and then it's dephosphorylated. We see a similar pattern in the area postrema. Note the scale's different here, because vagal afferents also innervate this circumventricular organ adjacent to the nucleus of the solitary tract. Now, if you block, I should say, if you block the extracellular receptor kinase that phosphorylates synapsin, you don't see this increase in synapsin phosphorylation in response to CCK, and you attenuate reduction of food intake by CCK. On the other side of the equation, if you prevent dephosphorylation of synapsin, here with the calcineurin inhibitor, what you see is that you enhance the amount of phosphorylated synapsin that's produced in response to CCK in the vagal afferent terminals. And if you look at, and you extend the length of time that it's phosphorylated, and I don't have the full graph here, but synapsin in the presence of the phosphatase inhibitor, phosphorylase inhibitor, is extended out here to about 2 hours. And what you can see here is that CCK reduces food intake normally for a short period of time, over about 30 minutes, but in the presence of inhibiting synapsin dephosphorylation, you extend and enhance the ability of CCK to reduce the meal size. So at this point, I just summarize for a moment, then point out that reduction of food intake is by CCK involves activation of glutamate receptors in the hindbrain. It increases glutamate release from vagal afferent endings and triggers an increase in the available synaptic vesicle pool for transmitter release. And reduction of food intake by CCK can be prevented by inhibiting any of these processes, but specifically by inhibiting the phosphorylation of synapsin and enhanced by enhancing the dephosphorylation. So we don't know at this point whether other gastrointestinal peptides may share some of this action. This is an area of great interest to us. And we also don't know about the possibility of synapsin phosphorylation or other components of the synaptic processes that may be involved in modulating transmitter release. And I'll deal with this also a little bit later in the talk. So up to this point, we've talked a bit about, or I've talked a little bit about, the control of food intake by signals from the gastrointestinal tract, and that's one of the things that Tim also emphasized. And that is that the gastrointestinal tract is the part of our physiology which provides signals that are directly related to food that we've just eaten, that's in the process of being digested and absorbed, the quality and the quantity of that food. Whereas other compartments of physiology of an organism provide indirect information on catabolism of glucose or of carbohydrates, proteins, and fats. And those signals we know provide information that's used to control food intake. And interestingly, of course, the white adipose tissue also provides endocrine signals that affect food intake, notably leptin, the adipokine that's produced by white adipose tissue, and acts on the brain and reduces food intake. If we look at the way in which a signal like leptin reduces food intake, it's very instructive and interesting. Injection of leptin dramatically reduces food intake and can do so over days and longer periods of time, leading to reductions in body weight. And if you look at the meal parameters here, what you see is that reduction is virtually entirely due to reduced meal size. And so it appears that this non-gastrointestinal protein signal that controls food intake is producing an effect on meal parameters very similar to the feedback that we get from the gastrointestinal tract, and specifically from gastrointestinal hormones like cholecystokinin. And so it seems important to know whether vagal afferent function is modulated by signals like leptin, and I'm going to try to address that question a little bit, focusing on vagal afferent synaptic transmission modulation by melanocortin receptor activation. Now, why I focus on that will come up in just a minute. But I want to first mention that the interaction between gut hormones and between gut hormones and leptins begins in the gastrointestinal tract and begins at the peripheral vagal afferents. About 80% of vagal afferents that innervate the stomach and small intestine have CCK receptors, and about 45% of them express both CCK and leptin receptors. So both receptors are there, and if you infuse leptin and CCK intraarterially so that their action is limited to the gastrointestinal vagal afferents, or the area that they innervate, you can show that leptin and CCK enhance each other's action. And here, the combination of subthreshold doses of leptin and CCK intraarterially administered reduce meal size, whereas those concentrations are ineffective in the systemic circulation. So the potential is there for altering the meal by peripheral action. But most people would say that leptin's most dramatic action in control of food intake occurs when it acts on the hypothalamus, where it activates pro-opiomelanocortin neurons, POMC neurons, to release alpha MSH and inhibits MPY neurons. And but this effect is supported by the fact that leptin does activate the POMC neurons. Alpha MSH acting at MC4 receptors does reduce food intake, and it does so by reducing meal size, and antagonists of MC4 receptors attenuate leptin-induced reduction of food intake. So there's almost completely. So there's good evidence that this mechanism is working. And I might also add that there's an interesting connection between the MC4 receptor and CCK-induced reduction of food intake, and that is that MC4 receptor antagonists administered to the hindbrain attenuate the response to CCK. So an interesting experiment several years ago was done by Zhang et al. In collaboration of Hans Bertud and Harvey Grill. And what they did was to inject leptin into the arcuate nucleus of the hypothalamus and look at reduction of food intake. And then they injected a melanocortin antagonist, an MC4 receptor antagonist, into the hindbrain. And what they showed is that leptin indeed in the arcuate reduced food intake by reducing meal size, and that effect was blocked by this antagonist of the MC4 receptor, SHU9119, injected into the hindbrain. And this suggested that perhaps there's a modulation of what's going on in the hindbrain by forebrain POMC neuron activation. We've postulated that effect occurs actually at the vagal afferent terminal itself. And some of the evidence for that is that MC4 receptor is expressed by vagal afferents. And if you look at labeled vagal afferents here in red, they are closely applied to MSH, immunoreactive POMC fibers in the hindbrain, so they have an opportunity to react together. And so we would suggest that POMC neurons modulate vagal afferents by acting at receptors at this first presynaptic element in the visceral afferent communication pathway. And indeed, that seems to be happening. What I'm showing you here is, again, phosphorylation of synapsin, this time mediated by protein kinase A, which is activated by the MC4 receptor. And it phosphorylates synapsin at serine 9. And this stack of images just illustrates the co-localization of phosphorylated synapsin and vagal afferent neurons. And you can see here that MC4 administered into the MC4 agonist administered into the hindbrain elevates synapsin phosphorylation for hours after it's administered. The effect of this phosphorylation is similar to what I've already shown you for the ERK phosphorylation of synapsin, but perhaps even more potent. It enhances transmitter release. And if we block the phosphorylation of synapsin with a protein kinase A inhibitor, we block the phosphorylation of synapsin and we reverse reduction of food intake by the MC4 agonist. So what's this doing? It's actually enhancing vagal afferent transmission. And for a variety of reasons, which I won't have time to talk about, we know that's the case, but here's some recent electrophysiology. And what you're seeing here is, I'll only talk about a couple panels, is a % failure rate of impulses coming into the hindbrain in this slice preparation, like I showed you earlier. And you can see that very quickly during a bout of stimulation, which is within the physiological range, there's a 70% failure rate of transmission from the vagal afferents to the NTS. And activation of the MC4 receptor cuts that rate in half. It also decreases the rate of decline of the postsynaptic depolarizations that we see, the excitatory postsynaptic currents that occur in response to vagal stimulation. So what we're doing here is increasing the fidelity and strength of vagal afferent transmission in response to the MC4 activation. And so what I'd like to propose is a model that needs further study, and that is that CCK and other gut peptides activate vagal afferents and provide a primary signal for satiation. that signal is modulated by signals such as leptin and perhaps other endocrine signals that activate receptors on the vagal afferents, such as the alpha MSH receptor, the MC4 receptor, and doing so enable, they're gatekeepers that enable the vagal afferent to faithfully communicate and amplify the signals coming in from the gastrointestinal tract. And in that way, this vagal afferent ending can be viewed more or less as a paintbrush that paints the feeling of satiety, the sensory process of satiety, satiation, on the hindbrain. And you can hold the brush back here with CCK and gastrointestinal inputs, or you can hold it close to the bristles here with MC4 receptors and get that satiation experience started. I think I'm running out of time, and but what I want to leave you with is the idea that gut peptides, gastrointestinal peptides, as well as non-GI hormones, interact. And one of the places they do that is in the nucleus of the solitary tract, at the first synapse, the first visceral afferent synapse, where the experience of satiation begins. And that this is an important place where food intake is monitored. And I, like everyone else, I'd want to acknowledge that if you're walking through a field, I'm a veterinarian, and so I do that sometimes, and you find a turtle up on a post, you know that he had some help getting there, probably. And I'm fortunate to have had a lot of help. And I especially would acknowledge my former student, Carlos Campos, and colleagues James Peters and Susan Appleyard in our department, as well as other muses, and my support from NIDDK and NINDS. Thank you very much.